Hi everyone, my name is Joshua Liu, and today I'll be talking about the effects of glyphosate on algal health in Orange County coastal waters. So firstly, what is glyphosate? Glyphosate is an active component in many herbicides, a popular one being Roundup weed killer, that is widely used commercially and privately. These herbicides are intended to only target weeds, however glyphosate acts as a non-selective herbicide, meaning that it has an effect on all plants. This is because glyphosate targets the shikimate pathway, which is a metabolic pathway involving amino acid synthesis in many organisms, including not only plants, but algae as well. Glyphosate has also been found to last for up to a year after introduction into saltwater environments. Because of this non-selective quality of glyphosate, the fact that it targets an integral part of photosynthesis, and its ability to remain in saltwater systems, it's really important to investigate the effects that glyphosate has on the ecosystem. With mass urbanization in Southern California, there's increased runoff to coastal ecosystems from the upkeep of lawns and contact with contaminated surfaces. Study of the effects of glyphosate on algae is crucial because it's abundant in the rocky intertidal ecosystems of Southern California. There are limited methods to effectively measure glyphosate and studies on its effect on the ecosystem, especially in saltwater ecosystems. And there are a few studies on how glyphosate impacts algae found in the rocky intertidal. In our field site Little Colonel Del Mar, you can see many mansions on the cliffs overlooking the beach. There's plenty of runoff of herbicides occurring to upkeep their lawns, as well as additional wastewater from the city that collects into man-made creeks or drains that are placed along the shore. These creeks or drains put out water directly into the ocean or onto the shore, where the water then runs through the tide pools. Because of the nature of tides and tide pools, water can be held in tide pools during low tide for prolonged periods of time. Tide pools that are high enough on the shore may not make contact with the ocean for even longer periods of time. The unpredictability of runoff, in that the concentrations of glyphosate can vary widely day to day, and being in tide pools can cause algae in the intertidal to be subjected to various concentrations of glyphosate for varying periods of time. During the summer, we investigated LC50 trial techniques and collected information on glyphosate from the field. Firstly, in order to find the concentration of glyphosate in a sample, we have to transform it to phosphate. This is because we use the latchet, which uses color spectrophotometry to determine the concentration of nitrate, nitrite, and phosphate in water samples. The latchet can't detect glyphosate in its raw form, so we used a method to convert glyphosate to phosphate developed in the lab previously that involves boiling the sample down to a solid and adding hydrochloric acid. To test the conversion of glyphosate to phosphate, we used standards of glyphosate, and we found that there is an over 95% conversion, a value that was above our expectations because an approximate 95% efficiency is normal for the latchet. One factor that needs to be accounted for is the fact that phosphate survives the transformation process. Therefore, to get the actual concentration of glyphosate, measurements need to be taken twice on the latchet, pre-boiling and post-boiling, where the difference between pre- and post-boiling is the actual concentration of glyphosate in the sample. This bar graph shows the concentration of glyphosate at various locations in our field, with the EPA safe drinking water limit for glyphosate shown at 0.7 milligrams per liter or 4.1 micromoles per liter. It was found that there was higher levels of glyphosate in the man-made creeks and drain, Buck Gully, Morning Canyon, and the storm drain, and there were lower levels in the polluted pools, which are tide pools that are directly situated below the storm drains. We determined that this could be due to tidal activity that flushes out the tide pools and recycles the water. There was also no glyphosate found in the ocean samples, which we believe is due to the glyphosate being heavily diluted. With our previous research and findings from the summer up until now, we plan on performing LC50 trials on 3 different types of algae, where the LC50 is the concentration of glyphosate, at which 50% of the algae are dead. We will also investigate the photosynthetic efficiency of the algae in the field, as well as in response to varying concentrations of glyphosate in the LC50 trials. Finally, we'll look at the concentrations of glyphosate in the field and of each replicate in the LC50 trials at specific intervals. This will all help us to investigate the relationship between glyphosate concentration and photosynthetic efficiency of algae. We hypothesized that glyphosate at concentrations of 50 micromoles per liter or higher will have considerable negative effects on photosynthetic efficiency of macroalgae in the rocky intertidal of Little Coronel Del Mar. We also hypothesized that Ovalactuca, a green algae we'll be looking at, will experience the greatest decline in photosynthetic efficiency of the 3 algae due to the fact that it's more delicate and plant-like than the other algae. We plan to use 3 types of algae that are common in our field site: Sargassum horneri, which is an invasive brown algae, coralline, a red algae of the subfamily Corallinoideae, and Ovalactuca, a green algae. To perform the LC50 trials, we'll first collect the algae from our field site. Using previous literature and our findings of the concentrations of glyphosate in our field, we'll set up 15 1-liter containers of artificial seawater with increasing concentrations of glyphosate, where each concentration will have 3 replicates. These will then be placed into a growth chamber for 4 days at a time, with a day-night cycle and temperatures that mimic the conditions of our field. Twice daily, we'll use a pulse amplitude modulated fluorometer on the algae. Then we'll extract chlorophyll from the samples post-experiment. To look at photosynthetic efficiency, we explored pulse amplitude modulated, or PAM fluorometry. PAM fluorometry is used to measure the photosynthetic efficiency of photosystem II, which is the first protein complex in light-dependent photosynthetic reactions. With this method, varying levels of light are used to form light curves of different data. The three values we'll be looking at are ETR, or electron transport rate, Y2, which is the yield of photosystem II, and FV over FM, the max efficiency of PS2, which are all ways of gauging the photosynthetic efficiency. During the trials, we want to see how the algae reacts under different conditions, varying concentrations of glyphosate, and how its photosynthetic activity is affected by it. PAM fluorometry will use varying levels of light and track the three previously discussed parameters, which will tell us the upper limits of its productivity, as well as changes in efficiency over the duration of the LC50 trials. This graph shows a PAM light curve, which shows Y2 and ETR over different light intensities on algae. Using the graph, you can interpret a higher photosynthetic efficiency with higher values of Y2 and ETR. This algae was freshly taken from the field before a glyphosate treatment. Taking light curves of algae before the LC50 trials can help us establish a baseline of the curves that the untreated algae may follow, which we can use to compare to glyphosate-treated algae. In terms of data analysis, we'll use ANCOVA to account for the initial PAM values on algae fresh from the field. We'll also use ANOVA to compare PAM values between concentrations and trials, and linear models to explore the relationship between glyphosate and photosynthetic efficiency. Thank you so much for listening, and I'd like to thank UROP for this opportunity, Rachel Hill and Dr. Matthew Bracken for their mentorship, and the Bracken Lab as a whole for all the support.